Now my pleasure to introduce the next speaker, Professor Tony Burkitt, who was actually an undergraduate here at ANU and a doctoral student in Edinburgh, and has been at the University of Melbourne for some time, presently chair in biosignals and biosystems in the Department of Electrical and Electronic Engineering, and is the director of Bionic Vision Australia. Professor Burkitt has worked in a number of areas of medical bionics, including cochlear implant speech processing, neuroengineering, computational neuroscience, and biosignal processing for epilepsy. He'll be speaking to us about the challenge of restoring sight with vision prosthesis. The title is R&D in Visual Prosthetic Devices, Recent Progress. Tony.
Good, thanks very much for that introduction. It's been my great privilege to lead a very exciting program and group of researchers. There's a project funded by the Australian Research Council through a special initiative to develop prostheses for vision impairment. It was funded by the Australian Research Council. The partners in this, our members, two universities, the University of Melbourne, the University of New South Wales, two medical research institutes, the Bionics Institute, and the Centre for Eye Research Australia, and NICTA, National ICT Australia, and we're working particularly with the labs in both Melbourne and in Canberra. Our partners in this have been the National Vision Research Institute, based in Melbourne, University of Western Sydney, and the Royal Victorian Eye&E Hospital, which is where our clinical and surgical team have been housed. It is a very large research project, so I won't have time to talk to you about all of it. What I want to do today is to really focus upon the part that's now moving forward into clinical and commercial translation. We have had 3 patients who have been implanted with a prototype device, so I'd like to be able to show you some of the results of that study and explain a little bit about where we're heading with the commercialization of that work. The program has been focused, as I was saying, upon the development of retinal devices to restore vision to the blind. Just to take you through some of the basics about how the whole implant system works, many of you will be familiar with how a cochlear implant system works, and so you'll recognize many of the same components that I'll be talking about here. To gather the external signal, we have a camera situated on a set of glasses, but this image needs to undergo quite a lot of processing before it's suitable to be used for the implant system. So that signal is collected by that camera and goes then to a body-worn unit, something initially that will be around about the size of an iPhone, to be carried sort of in somebody's hip pocket, that transforms that signal into the form of electrical impulses that can then be used and transmitted together, both the signal and the power transmitted through the skin, through an inductive coil, in the same way as done with a cochlear implant, to a processor that's implanted behind the ear. The signal is then decoded and sent down through a lead wire system to the implanted components at the back of the eye. And I'll show you a close-up view of that in a moment. But the electrodes there are close enough to the surviving nerve cells in the retina, so it sends out the electrical impulses picked up by the surviving nerve cells, and that signal is then sent to the vision processing centers of the brain, where it's then interpreted as a vision. Somewhat different to what those of us who have normal vision, but we'll talk through some of the differences of that and how the patients experience that. This is what a healthy retina looks like. So there's an image taken through the front of the eye, and you can see the central part of the retina here, the macula, which has the high concentration of the photoreceptors for the normal vision. You can see the blood vessels here, and of course they leave the eye at the optic nerve head. Now, for a normal retina, the light comes through from this side and travels through the different layers of the retina through to the rods and cones, the photoreceptors, which are situated right at the very back of the retina. And these transduce that signal into the electrical impulses. Quite a lot of processing actually happens within the retina before the signal is then transmitted through the retinal ganglion cells out via the optic nerve through the to the vision processing centers of the brain. So in a sense, it's the activity of these retinal ganglion cells which is the important output of the eye. And so long as these cells are functioning, they can become the targets then for the electrical stimulation for our device. What happens for people with retinal degeneration is quite typically that it's the rods and the cones, the photoreceptors that have died off, but leaving these other cells there to become still viable, but of course not receiving the signal from light, but can then receive an implanted signal can actually still stimulate these cells. So in healthy vision, we can get a very, very high acuity transmission of the signal right through from the nerve cells in the eye through the vision processing centers of the brain. But for conditions that involve some sort of degeneration, and our main target in the first instance will be people who have retinitis pigmentosa, and you can see through this image here the degeneration corresponds to the darker patches of the retina here. And for a condition like retinitis pigmentosa, this results in the loss of the peripheral vision initially. People end up with these patches of vision, almost a tunnel type vision, in the very center of the visual field. And in the later stages of the disease, that disappears entirely. Retinitis pigmentosa is the leading cause of inherited blindness. It affects roughly 1.5 million people worldwide, and about a quarter of these patients are legally blind. It typically has an onset when people are adults, so late teens, early adulthood, and quite often by the time people reach middle age, they're legally blind or have lost their vision entirely. Eventually, we would like this technology to also be able to be used for people with other conditions. The most prevalent one of these would be age-related macular degeneration. And this is a condition that's affecting increasing numbers of people, particularly in Western countries, as our population ages. It has a very different disease progression, typically affecting the central part of the visual field. So if you're looking through the front of the eye, as in this image here, you can see the degeneration and discoloration right in the central part of the visual field. In Australia, it costs more than $2.5 billion per year. Affects mostly people aged over 65, but again, increasing number of people as our population ages. This is becoming more and more important as a disease to be treated. So what will somebody with a retinal implant see? Just to give you an idea of the sort of technology that we're talking about and where we're heading with this implant technology, I've just taken here a typical sort of street scene. And if we think of each of the electrodes in the eye as providing, as it were, a pixel of vision, this gives you a sort of visualization of the type of information that we can hope to produce as the technology increases. So with a very small number of electrodes, this is of the order of 16 pixels for this particular picture, you only get very, very rough information. But as the number of pixels or the number of electrodes increases, you get increasing information from the image. The number of groups around the world who are working on this technology, and we're all sort of in around about this sort of area here of tens of electrodes, so providing information that's of the order of sort of tens of pixels at the moment. Now, for somebody who is completely blind, this still provides an enormous amount of information. I mean, what you see here is, of course, a static image. The people who are using this technology have the camera attached to their glasses, and so they're actually able to move their head, and they get a lot of additional information through being able to do things like scan images and pick up edges and that sort of thing. We do hope, though, that over the coming years we will be able to get a larger density of electrodes, eventually moving up to 100 and even beyond that in future generations. But that will take many, many years of development to get to that point. So what is it that people actually report as seeing with this electrical stimulation? I've mentioned that we've taken the visual input and we've transformed that through a number of stages involving the manipulation of that image into a series of electrical impulses. And now what we want to do is to choose a strategy, that sort of transformation of that image into electrical impulses that gives the best benefit to the patients. And we measure that by doing psychophysical experiments with the patients to determine what features of that visual information they're actually using when they perceive the image. I should say that all of the electrical stimulation is done with these biphasic pulses. The reason for it to be biphasic is we don't want to leave any residual charge in the eye that actually does damage to the tissue. So we have biphasic pulses that are stimulating the neurons there. And in the simplest sort of naive stimulation strategy, each of the pixels from the original image correspond to one electrode, and the intensity of the image from that pixel will determine things that we can adjust in electrical stimulation, like the pulse amplitude, the pulse frequency, the pulse width, these sort of things. So that's the very sort of simplest naive strategy, and there's clearly a lot more that goes in behind all of that to the signal processing that determines how that's really used in practice. But what people actually perceive are things called phosphenes. So these are flashes of light in the visual field. They can be both white or dark. So we have the ability to, in a sense, with our visual field, to detect changes away from the normal level. There's always a residual level of activation in the visual system. So patients actually do report seeing both light and dark phosphenes with the electrical stimulation. Now, with a very small number of electrodes, you only get a very coarse representation. But as you go up, and this is now just an image that's been transformed or pixelated, or in this case phosphonated, to give you an idea of what a typical sort of street scene would look like as you increase the number of phosphenes. This is going up to the sort of level tens of phosphenes. By the time we get up to about 1,000, then we've got a fairly high acuity version of that picture. And of course, that's the original picture. Within our program, we've had a series of generations of the technology that we've been working on. The very first stage of this has been a prototype device that involves 24 channels. We call this the wide view device, and the object of this technology is to provide patients with the ability to be able to move around in their environment. So it's not giving them that very high acuity vision that they'd need to read print or do that sort of thing, but it's providing them with the vision to be able to avoid obstacles, to be able to find where there are doors, windows, to give them back independence in their everyday life. So this is one that has been developed at the Bionics Institute and through our collaborators in Melbourne. The second version of that is a 98-channel device that has been developed at the University of New South Wales, but both of these using the sort of materials that we're very confident with from cochlear implants, platinum and silicone. And then there's been also at the University of Melbourne and collaborators in Melbourne a high acuity device that's based on a quite different technology, a diamond-based, very exciting work that has huge applications in other areas. I won't get an opportunity in this short talk to talk about that technology, but it is very exciting, but still in preclinical work. What I want to do here is to really talk you through the studies that we've done with our first 3 patients with our prototype device. Like I say, this is initially a 24-channel device, and we're currently now developing that, expanding the number of electrodes. The next stage of that will be a 44-channel device that we're seeking to take through to commercialization. This image here, if you actually look very carefully, you can see the implant, the part that actually goes inside the eye here is this part component down here. There's a number of smaller platinum electrodes at this end of the electrode array. These 2 large dots that you see here are actually on the back of the array, and these are the return electrodes where the current is then collected and returned back to the stimulator. As part of the program, one of the first things we did was work with our surgical team to develop an implantation technique to be able to place the implant into the eye using a surgical procedure that we'd developed that hadn't been used anywhere else in the world. It's called a suprachoroidal implant position, and this shows a cutaway through the eye, showing the external part here, the sclera. Just inside the sclera, there's a series of blood vessels called the choroid, and it turns out, and inside of that, of course, is the retina itself. It turns out that there actually is a small space there that can be opened up surgically to implant the electrode array, and that's the position that we're using. It turns out to be extremely stable from a mechanical point of view. In patients who have these sort of forms of retinal degeneration, the choroid is typically very thin, so in fact the choroid only presents a very small barrier to the electrical stimulation that's going through to the surviving nerve cells. This shows just some of the details of the development of that surgical approach, and it can be done relatively straightforwardly. I'm told by my surgical colleagues that in fact anyone who's trained in eye surgery would be able to do this technique, and it typically only takes them 2 to 3 hours with any of the patients that they've done. As part of our program, the very first thing that we needed to do was to establish the safety of the device. So safety followed by efficacy were in fact what we concentrated on in the early stages of the program. And so a lot of this work, particularly for this device, has been done at the Bionics Institute, and I won't have time here to talk about the very extensive studies that were done involving analysis of histology, first in acute studies and then over chronic time periods of 3 to 6 months, to make sure that we weren't getting any inflammatory response in the eye. We've also done studies to show that this device could actually be removed. So if there's a need for infection or any other reason to actually remove the device, we've shown that it can be removed safely, and even with animal studies, if you remove the device, it is possible to actually re-implant the device. So that provides us with a lot of confidence going forward with our clinical studies. We were extremely fortunate to have 3 of our first patients, all incredibly altruistic people, and really they've become part of the research team. We owe an immense sort of gratitude to them for the time that they've spent working with us. Initially, the study was an 18-month study. It was never envisaged to be a permanent implant for these particular prototype patients. In fact, that was extended to 2 years, and they would come in, each of them, for a day or more per week into our testing facilities for psychophysics at the Bionics Institute and for the clinical assessment at the Royal Victorian Eye and Ear Hospital. In a sense, they've become also our greatest advocates. I've mentioned that the surgeries were all done at the Royal Victorian Eye and Ear Hospital, and we're extremely grateful to them for their support. Tremendous place to be able to do this study, because of course that was also the first hospital where the cochlear implant was done in Australia. So they've got an enormous amount of expertise and experience with this technology. This shows a close-up of the electrode array itself. You can see the smaller dots down here are the stimulating electrodes collected with each with a platinum wire that goes through the lead wire system here. So an enormous amount of work goes into developing the mechanics and the shape and stability of all of that, and the 2 return electrodes, the 2 larger dots you see there. I've mentioned all the materials, the ones that we're very, very confident with in terms of their safety profile. And all 3 patients had retinitis pigmentosa. We had to be very conservative in our patient choice, because we had to only choose patients who really had no functional vision, had profound vision loss, and we weren't putting at risk any residual vision that they would have had. So these were patients who had the very far spectrum of vision loss. And I've mentioned previously, they were originally implanted for 18 months, but that was then extended to 24 months. This shows the full system here. For this prototype study, there were no implanted electronics, so we didn't put the chip or the stimulator actually implanted in the patients. They were connected through a percutaneous connector that you can see here, and they were connected only when they came into the testing facility initially, and then we actually had then a semi-portable device that we could test them as they were moving about in a controlled environment. This shows a view of the testing facility at the Bionics Institute. There's actually one of our graduate students demonstrating it here with the different components to do with the stimulation and the switching of the array. But also here you can see eye trackers. We wanted to be able to determine exactly where it was that the patients were experiencing the stimulation and what they were seeing, which part of the visual field. So they could actually point to this, and this was measured here with this finger tracker. They could describe exactly what they were seeing, and we could use that then to fine-tune the electrical stimulation. And that's played an enormous role in us going forward to be able to develop the vision processing for our next generation of devices. The first switch on happened in July 2012. It was a very exciting day. I mean, we were sure at this stage of the safety of the device, and we were fairly sure that the patients were going to be able to see something. We had absolutely no idea how it would work. So it was a very exciting day, and I think that the smile on Di's face sort of says it all when we got that first indication that she was in fact seeing the phosphenes and could describe them in extraordinary detail. And you see her here using the full system to sort of sketch out exactly what she's seeing with various forms of stimulation. And this has played an enormous role in us going forward, developing the sorts of stimulation strategies that we need to provide functional vision for these patients. We're also, of course, monitoring it from a clinical point of view, and you can see here this is now a view through the front of the eye. The outline here is of the electrode array, and the individual dots are the electrodes. And so we can monitor this over the course of the implantation and show that there's been no movement of the electrode array during the course of the study, and we can also monitor the health of the retina. This shows an OCT image here, and you can see the different layers of the retina here. That's the dark part is the electrode array itself, with the retina slightly displaced in front of that, but just showing very, very good health over the entire course of the implantation. And even closer view, you can, if you look very carefully here, you can actually see the individual platinum electrodes in the electrode array situated here with the retina on top. So it's these sort of very, very high quality images of the health of the retina which provided us a lot of confidence going forward with the device. And we've been able to test a lot of different sort of stimulation modes. I won't go through these in any sort of detail, but this gives you some idea of the sorts of things that we've been testing out. So the red here represents where we do the stimulation, and the return electrodes are the ones in black. So we're testing out different types of what we call monopolar, bipolar, common ground, tripolar, all sorts of stimulation techniques to be able to assess what it is that the patients actually perceive in terms of the size of the percept, the intensity of the percept, the localization of it, and so forth. And that's been very important for us going forward. In terms of the clinical assessment, I mentioned that our patients all had profound vision loss, and that's actually defined as less than 6/120 or 2400. In fact, our patients were at the very far end of that scale, so in fact much worse than that. And all of our patients only had bare light perception. They could only barely tell whether in fact it was night or day. What we found through the course of the study was that their vision did fluctuate, that there were enormous differences. As we were working with them over this extended period of time, we could see that there was enormous differences in their vision ability, and that all of these standard visual acuity tests actually just couldn't be used at all. We actually had to develop a whole set of new testing abilities, really test themselves to work out what it was that these patients were seeing, and to be able to work out what sort of visual acuity that corresponded to, and how that could then be improved for use in everyday life. So the first part of the study was done entirely within that testing facility. We then developed a semi-portable version of the processor, and you can see here, this is now with one of our other subjects, with the camera here, the eye tracker still there to be able to continue to monitor that. The backpack contained all of the electronics with the algorithms and things that we're testing out, and the battery packs. And so this enabled us to then test the patients in a more sort of everyday environment, initially in the hospital, but then also taking them out onto the street. So a lot of the benefit of these devices, though, is really the question of how people can use it in their everyday life. And so we developed a whole series of sort of real-world measures to assess how the patients were using the technology, how they were using it for orientation, mobility, and for activities of daily living. And that's really helped us to inform both the device itself, but also the sorts of rehabilitation and training that's needed for the future for these patients. As part of this, we'd set up a testing facility actually here at the NICTA Labs in Canberra. And this was using a particular form of vision processing that we call depth processing. So this is where, rather than encoding the intensity of the image in terms of the intensity of rather than encoding with the electrical stimulation the intensity of the image, what we did is we encoded the depth of the image. Right? Now, the reason for doing this is that the most important thing for patients is actually obstacles that are nearby. Things that are much further away are much less sort of salient to the patients. They really want to know what it is they're going to be able to trip over or where they find things that are nearby to them. So if we can actually encode things according to how far they are away, it becomes much more useful to the patients. And so to test out ideas like this, we set up this testing facility. And this is Nick Barnes, one of our chief investigators who is leading the vision processing part of the work. And he explains us taking the patients through this testing facility.
So today Diane's doing experiments where we're having a set of obstacles in front of her on the ground, and she needs to move from the back part of this environment through to the front part, and we're looking at how long it takes her to pass through that pass and how many collisions she has with obstacles or brushes against the wall. So she's wearing a backpack computer which is attached to a head-mounted camera incorporating a depth sensor, and the backpack is processing that and then doing the vision processing of that and then putting up the set of things that we want to have stimulated on the electrode array for her to then gain an impression of what she's seeing in the image.
Oh, this is, you can see a reason for the bionic eye. You can see that there's a practical use for it. It's just, it's really good. Well, it's different sight than what I had before I lost my vision. So it's learning to trust what I'm seeing, and that's why I say the more I'm doing it, the more I'm trusting what I'm seeing and learning to see what I'm seeing is true. That's what's there. The more I'm doing this, the more I feel like I'm looking out of my eye. So I know there's a camera there, and
Good. So I think it's very exciting for us to be able to get that experience of the patients, for them to be able to feed back to us exactly what it is that they're seeing and how we can then incorporate that into our next stage of our development of our project. So shown here is the image for where we're going at the next stage, which is using in fact it is actually the same footprint of the device, so the device itself hasn't actually got any larger, but we have incorporated now 44 electrodes, so almost doubling the number of electrodes onto that device, and it'll have much more sophisticated signal processing. And with this next generation of the device, it will, like the cochlear implant, involve all of the implanted components in the behind the ear unit. So the data and the power will be transmitted from the external processor, be decoded on the internal components, and that device will be then for the lifetime of those patients. We have funding initially from the NHMRC to do a small-scale clinical trial, which is starting this year, and we're just currently in negotiations with investors to then carry that device forward into a larger-scale clinical trial and eventually into commercialization. So we're hopeful that there will be an announcement over the coming weeks and months about that. So it's a very large project. And it's not possible to list all of the investigators here, but we are very grateful to all the teams that are involved. Part of the great sort of honour of being the director of this is to be able to see all the work that's been going on. We've been very glad to also develop our next generation of students. We had an enormous number of students, PhD students, early career researchers associated with the program. Nick, of course, is one of my own PhD students, so very grateful to see that. As well as the work that's been going on in all of the different labs, and we'll hear some of the other areas in which people are doing bionics. Of course, some of the work being done at the Bionics Institute, and Rob will talk about some of the other work in other non-eye related areas, as will Michael at the National Vision Research Institute today. But thank you very much for your attention. I'll be happy to take questions.
Thank you, Tony. I think Srini had his arm up first, and then you have, I can't remember your name. Sorry.
Thank you very much, Tony. Brilliant talk. Thank you so much. I had a question about the simulation that you showed with the I guess the 1,000 electrode array, and when you showed that image and then showed us what it really was, which is a picture of a tram, I could barely believe it. And it looked so different, right? I wouldn't have guessed it was a tram. And I wonder whether the reason why it works so well is because there's a fair amount of spread in the activity from each electrode. So it's really what the patients are looking at is a low-pass filtered version of that punctate array. And so it's really like looking at a real image through a defocused lens of a camera or something, right? It's a blurry version of...
Yes. In fact, you put your finger on one of the key sort of issues that we're working with in terms of stimulation strategy, is to try and control where the current actually flows in the eye. So this question of current flow and current localization and localization of the activity in the retina is one of the most active in this whole field. I mean, not just with retinal implants, but more generally for any stimulating device. But absolutely, that's one of the key determinants of the eventual visual acuity that patients will get.
Tony, it's really exciting to see how far this has progressed. You didn't put much time into telling us about the processor, and I'm curious to know, given that in normal vision, as the visual field moves across the retina, the retinal ganglion cells do rather sophisticated things, on responses, off responses, directional selectivity. Can any of those features be ultimately embedded into the processor?
We're working actually on a number of things to try and do that. I mean, one of the ones you actually put your finger on, that sort of difference between on and off, and we've been investigating, in fact, in Michael's labs, some of the stimulation strategies that can preferentially stimulate different types of retinal ganglion cells in order to be able to use more of that circuitry that actually exists in both the retina and actually in the further vision processing pathways in the brain. We're still at the very early stages of doing that, so we do have some stimulation strategies that work in the lab to do that, but we haven't yet been able to test those out in patients. We hope that as we get more and more patients and we get more ability to be able to test out exactly those sort of strategies to do those sort of things.
Thanks for that, Tony, up here. I take it the cameras are currently visible light cameras. Have you considered using other kinds, like infrared, which would give people with a bionic eye extra range of vision that people with normal sight don't have?
We haven't yet, but look, you're absolutely right. There's no reason why we couldn't. And in a sense, there's no reason why these people shouldn't be able to see, in the dark, really. If we could actually have any sort of sensor that can transform into the image, there's no reason why that couldn't be incorporated. In the sense, we've concentrated on the simplest, most straightforward thing first off, but you're quite right. There's no reason why that couldn't be incorporated in latter versions.
A couple of quick things. First is, can you comment on the impedance between the electrodes and the ganglion cells, given that the implant is not epiretinal? And my second question is related to the refresh rate of the electronics. As of now, I understand that the patients can distinguish between an object being there and not there, but eventually can they look at moving objects? Can they distinguish that? Thank you.
Yeah, so you put your finger on one of the really key sort of points about the technology. This electrode neural interface is one of the sort of key areas of scientific research associated with everything to do with bionics. And the more we understand about that electrode tissue interface, how that can be modulated through the nanoscale or macroscale structure of the electrodes themselves, the different materials that can be used, that's clearly going to play a more and more important role in the future of this whole field. it's an enormous area. I couldn't possibly do it justice in a couple of minutes, but it is a very, very active area of research. We've done some research in that area, but there's clearly an enormous amount more that can be done there. Your second point about motion and movement and the refresh rate, at this stage, I mean, one of the things we did at the very outset was to work out what was sort of the minimum refresh rate that we needed to do, and clearly in order to make sure that we don't get this sort of jerky vision, we want to make sure we've got a refresh rate of at least between 30 and 50 hertz. But we're now looking at much more sort of elaborate ways in which we can get smoother motion movement, even the ability to be able to, say, do the electrical stimulation in a way that preferentially enables the patients to pick up edges of objects and that sort of thing. That's all stuff that's actually being developed in terms of the research, and we hope eventually will then find its way into something that the patients use, but that's still for the future.
Good. Let's make this the last question. Thank you.
This is a really fascinating development, and I'm interested because we have these rods and cones. The rods are more sensitive for night vision, and cones are for the daily vision. But the electrical stimulation is activating primarily rods or cones.
Well, no, in fact, for all of our patients, the cones and the rods have all completely gone. I mean, they've had some sort of disease process, so the photoreceptors themselves are typically completely gone. The targets of the stimulation are ultimately the retinal ganglion cells, the output cells. We do also, as part of that, also stimulate other cells, like the bipolar cells, amacrine, all the other cells in the retina, but they're in fact a secondary sort of activation. The primary targets are the retinal ganglion cells.
And ganglion cells. So that at the end, these patients will never be able to see the world in color? Is that the...
It's unlikely with the present technology that they'll be able to do that until we could actually get the stimulation down at that sort of single retinal ganglion cell level, and that's a long, long way off into the future.
Okay, thank you.
Well, we've seen great progress in this group here, and we're going to hear progress from other groups, but please join me in thanking Tony for an outstanding lecture.
Thank you. Thanks very much.